Hi, everyone. So now that we have finished talking about what DNA is and how it replicates, we are going to move right into genes and how they move into proteins. So the overview of chapter 17 is the flow of genetic information can only move in one direction. We always start with DNA, then we have to move to RNA, and then finally we can move into proteins, which is kind of the gist of this chapter. So the information that is held within DNA is in the form of a specific sequence of nucleotides. This DNA sequence gets inherited by organisms from our parents, and it leads to specific traits being dictated through the synthesis of proteins. Proteins are the links between the genotype and the phenotype, so the genetic makeup and the physical expression of that trait. Gene expression is the process by which DNA directs protein synthesis includes two different stages, transcription and translation. So altogether, DNA to RNA to proteins is made up of three different steps, replication and then transcription translation. That's how we can end up with our little albino deer amongst a family of regular brown deer. Today we are talking about 17.1. Genes that are specifying for proteins via transcription and translation. So how was the fundamental relationship between genes and proteins officially discovered is kind of what we're talking about in this chapter. The evidence came from a study of metabolic defects. So in 1902, a British physician first suggested that genes dictated the phenotypes of individuals through enzymes that catalyze and speed up specific chemical reactions. He thought that symptoms of an inherited disease reflected an inability to synthesize a certain enzyme, which he found that linking genes to enzymes required an understanding that cells synthesize and degrade molecules in a series of steps called a metabolic pathway. Nutritional mutants, which this is kind of just a scientific inquiry. So George Beadle and Edward Tatum exposed bread mold to x-rays, creating mutants that were unable to survive on a minimal media. Using crosses, they and their co-workers identified three classes of this mutation. Each lacked a different enzyme that was necessary for the synthesizing of arginine, which is an amino acid. They developed one gene, one enzyme hypothesis, which stated that each gene dictates the production of one specific enzyme. So this was their experiment. Here, they grew the three different types of mutants, and they discovered those different genes that could only affect that specific enzyme. The products of gene expression. Some proteins aren't enzymes. In fact, so researchers later had to revive that one gene, one protein hypothesis. Many proteins are composed of several different polypeptides, each of which has its own gene that it codes for. Therefore, Beadle and Tatum's hypothesis is now restated as the one gene, one polypeptide hypothesis. Note that it is common to refer to genes and gene products as proteins rather than polypeptides, but it really is a polypeptide. They're not always proteins. The basic principles of transcription and translation. RNA is that first step. It's the bridge between genes and the proteins for which they code. Transcription is the process that synthesizes RNA using the information from DNA. Transcription produces first a messenger RNA molecule. Translation then reads that RNA molecule and produces a polypeptide using the information carried by messenger RNA. Ribosomes are the organelle inside cells in which translation is occurring. In prokaryotes, translation of messenger RNA can begin before transcription has even finished. But in a eukaryotic cell, since we are more complex and have many more organelles, the nuclear envelope separates the transcription and translation process. Eukaryotic RNA transcripts are modified through an RNA processing step in order for them to leave the nucleus and go into the ribosomes. A primary transcript is the initial RNA transcript from any gene prior to its processing. The central dogma is a concept that cells are governed by a cellular chain of command going from DNA to RNA to proteins, and they can only move in this direction. So this is our central dogma. We start with DNA in the nucleus. We make our RNA that can leave the nucleus that goes out into the ribosomes to become the proteins. Visually, in a bacterial cell, this can happen all at once, but we have replication and transcription that occur inside the nucleus, and then translation that happens out on our ribosomes. The genetic code. So how are the instructions for assembling amino acids into proteins encoded into our DNA? Well, there's actually 20 amino acids that can be made using our DNA. But there are only 4 nucleotide bases in DNA. So if you just think about that, there's only 4 combinations of nucleotide bases in DNA, but we can make 20 different amino acids from them. A codon is a triplet of nucleotides. So proteins and amino acids get read in something called a codon. It's a series of 3 nucleotides known as the triplet code. This is how we get from gene to protein based on that triplet code. It's a series of non-overlapping 3 nucleotides. The words of a gene are transcribed into complementary non-overlapping 3 nucleotide words of messenger RNA. These words are then translated into a chain of amino acids that forms a polypeptide. So here we have these 3 letters here, A, C, C. That would be a triplet code. Same, and then you would have the non-overlapping, so the next 3, A, C, C, G, A, G, T. Those are all codons, triplet codes. What you will notice, though, is when we get into messenger RNA, we're not binding with a T anymore. There is no T in messenger RNA. In fact, it's a U. Nothing else changes. The only difference is everywhere that an A would normally pair with a T, we're pairing with a U. And that helps us get to our amino acids. During transcription, one of the 2 DNA strands is called the template strand. It provides a template for the ordering sequence of the complementary strand of RNA during transcription. The template strand is always the same strand for a given gene. During translation, the messenger RNA bases form triplets called codons that are read in the 5' to 3' direction. Codons along a messenger RNA molecule are read by a translation machinery in 5' to 3' direction. So each codon specifies the specific amino acid, so there are 20 of them, to be placed in the corresponding position along the polypeptide. All 64 possible codons were deciphered in the mid-1960s. Of the 4 possible triplets, 61 of them code for amino acids, 3 code for the word stop, which is the end of translation. Every single protein will end with one of those 3 triplets that codes for stop. This is what tells the ribosome to stop making proteins. The genetic code is redundant, so more than one codon may specify for a particular amino acid, which basically is how we can go from 61 codons all the way to 20 amino acids. Codons do have to be read in the proper reading frame, so the correct reading groups, in order for the specific amino acid to be produced and the polypeptide produced at the very end. So these are all of your possible codons. I'm actually going to post a chart that's a little bit neater than this in Classroom for you guys. And it will give you the full name. So here you can see that there are 4 possible amino acids that code for leucine. You have your 3 stop codons right over here. Again, 4 that can code for arginine, but if you look, you have 2 more down here that code for arginine, so there's really 6 that code for arginine. But like I said, the chart that I'm going to put in Classroom is going to make a lot more sense than this one will. The evolution of the genetic code. So the genetic code is nearly universal, which means it is shared by almost every organism on Earth. The simplest form of bacteria to the most complex animals, we all share the same genetic code. Well, almost all of us. That means that genes can be transcribed and translated after being transplanted from one species to another. So there is actually experiments that take the tobacco plant that expresses a firefly gene, which makes that tobacco plant glow in the dark. And then there's also examples of pigs that have been expressing jellyfish gene, which makes part of them glow in the dark as well. There's a lot of genetic research behind this. Glow in the dark animals, for whatever reason, is becoming super popular, that we are studying those genes and the effects that they have on various different animals. All right, we are stopping here today. Thanks for hanging out with me and listening. It is part of your instructions for today. Make sure that you read chapter 17.1. You have taken notes. And then there is a really quick 5-question Google form for you guys to fill out. It is your technical assignment for today. As always, if you guys have questions, please go ahead and you may email me whenever you guys have questions, and I'll talk to you guys soon.